afternoon. My name is Ngoc. I'm PhD candidate of Hungarian University of Agriculture and Life Science. The former name Saint István University. I'm working in institutions of food science and technology. It is my delight to be here to present my topic in the biotechnology field. Under the name Screening of Cellulolytic Degrading Strains and Constructions of Consortium for the bio-pretreatment of wheat brands. Firstly, I will give some brief introductions of my topic and the research aim, followed by the experimental procedures and some dominant research will be presented also. Nowadays, our humans are facing global problems such as food shortage, environmental pollution, and energy crisis. The issues are becoming very serious, make it increasingly important to explore reasonable and effective ways to develop and utilize renewable resources. Lignocellulose is considered as a potential feedstock for production of biofuels and other bioproducts including chemicals, biofiber, biopulp, enzymes, huge amounts of sources from agricultural residues such as conserve, rice straw, wheat straw. The very cheap is abundant and co-friendly, so they can create a global priority. However, the main disadvantage of utilizing lignocellulosic materials lies in the crystalline nature of cellulose sheeting by hemicellulose, degree of polymerization, size, and bonding due to protected covering lignin with a low surface area for enzymatic hydrolysis. The reconstitution structures of lignin include the complex macromolecular structures, aromatic skeletons, varieties of functional groups, high energy density, and high molecular weight. Thus, to increase the digestibility of cellulose and hemicellulose, the removal or breakdown of lignin from lignocellulosic biomass is usually the target step of the pretreatment. There are various approaches for breaking down the reconstitution structures such as physical, chemical, and biological pretreatment. Before the saccharification and the fermentation of glucose into the variable product. Among various approaches, utilizations of cellulolytic microorganisms in bio-pretreatment has gained many interests due to the high growth rate of bacteria, low energy required, and eco-friendly. The selections and utilizations of synthesis microbial communities is challenging task to enhance the efficiency by bio-pretreatment. So, the purpose of the research is to characterize the potential cellulolytic degrading strains from Bacillus genus, and two main contents were conducted. First is the screening methodology from a strain of Bacillus bacteria, and constructions of a cellulolytic degrading consortium based on the high capacity of enzymatic production. In this research, wheat bran is used as the substrate for the biological pretreatment, and the basal medium were repaired with nutrient components such as lactose and minerals compound. pH-7 of the medium was obtained using the sodium hydride before autoclave, and the initial inoculum is 10⁵ cell per milliliter, and the incubation occurs under 140 row per meter agitations at 30 degrees Celsius. The microorganism is Bacillus with different species like Bacillus subtilis, Bacillus lingeniformis, Bacillus cereus, and Bacillus coagulans. The biological pretreatment were illustrated at the diagram. During the pre-treatment, the recalcitrant lignocellulose structure were broken down by the enzymatic activity of bacteria in submerged medium, resulting in solid residue and liquid hydrolysis. In the biological pre-treatment, the monocultures and the microbial consortium will follow similar procedure in other cultivations, and so different parameters were put into considerations. It is the weight loss, the reducing sugars, and the enzymatic activity. It includes different kinds of cellulolytic enzyme. Our work had gained many positive results. Shrinking the cellulolytic bacteria were conducted firstly to shrink the cellular degradation capacity of different bacillus bacteria. And you can see in the figure 1, the clear zones appear on the CMC agar plate. It proved that the presence of the endoglunacan, the dominant cellulolytic strain for the degradations. And to obtain fully understand about the effects of the enzymatic activity to the pre-treatment of which strain, more enzymatic assay will be present as follow. For the single cell cultivations, we can see in the figure 2 that's the Bacillus cereus, Bacillus coagulans, and has successfully degraded with overall higher reducing sugar content than the other two strains. And Bacillus subtilis B01162 placed at the second top among the efficiency strains. In order to elaborate the mechanism of the cellulose degradations, the enzymatic activity was assayed at 72 hours of harvesting. And so the total of 24 variances were classified into 3 clusters, including cluster A with Bacillus cereus and Bacillus coagulans, cluster B including Bacillus subtilis B01162, and cluster C for the remaining strains. Among the clusters, cluster A showed the highest enzyme activities, and it also had the high yields of fermented sugar among uninvestigated strains. And selenate activity of strain in cluster B is very less and remains at high level. However, the low reducing sugar were observed during the pre-treatment by the strain in the cluster B. And for the cluster C, the remaining strains were not as effective as other strains for single-cell cultivations. According to the screening experiment, 5 strains of different Bacillus species were selected for the construction of 11 different consortia. As you can see on the table below, the color cell indicates strains which were inoculated at 24 hours after all the strain cultivations. The enzymatic activities of the consortium AB, including Bacillus subtilis and Bacillus coagulans, by the time of incubation were decreased in the figure 3. As we can see in the figures, a longer pre-treatment resulted in the slightly enhancement of the total cellulose activity and selenase. In turn, the extracellulase endoglunacan activity dropped gradually during the time of the pre-treatment, and it remains at a very low level in the last 3 days. To discover the degradation mechanism of the microbial consortia, principal component analysis were employed to analyze the contributions of different cellulose enzymes in the first 3 days. The enzymes were measured, including the total cellulase, endoglucans, beta-glucosidase, and selenase. And 2 dominant components were extracted by this method. PC1 is accounted for 66.47%, and PC2 for an additional of 25.015% of the variability. The filter papers, or the total cellular activity, CMCs or endoglucanase activity, and selenase were positively explained by the PC1, while PC2 loaded with beta-glucosidase activity. The two-member consortia, including AB, AC, CD, and three-member consortia denoted as ABC, ABD, located at the right quadrant with high scores of cellulolytic enzyme activities, while the remaining six consortium took place in the other quadrants with lowest intensities of hydrolysis enzyme. We draw some important highlights for our research. The bacillus strain were able to grow and degrade with rain, releasing solute sugars. The member of bacteria consortia was selected based on the cluster analysis. Consortium of bacteria strains increased significantly cellulolytic activities. Efficacy of the consortium in with-bran pretreatment were increased significantly, and Bacillus consortia show an increase in cellular bioconversion than poor cell. And thank you so much for your kind attention.